I would like to invite all of the speakers, along with Adam Laing, who is the co-founder and chief scientific officer of IMU Biosciences, and Aleksej Zelezniak, who is the group lead at the Randall Center for Cell and Molecular Biophysics at King's College London, to the stage. We talked about a lot of really interesting omics technologies today, and a couple of things a couple of speakers said to me really resonated, which is number one, I think we are all really interested in the idea of being able to predict disease earlier. And then number two is that a lot of these technologies are complementary to each other. And so I'm going to direct my first question to Adam. I'm going to direct my first question to Adam, and actually, Adam, it'd be great also for you to tell us a little bit about IMU and what you're collecting, and just describe to us what do you think some of the gaps are in the data we're collecting, and how do you feel like IMU's platform is going to help those gaps?
Thank you, Melissa. So first of all, it's a pleasure to be here. My name's Adam Laing, and I'm a systems immunologist and the co-founder and chief scientific officer of IMU Biosciences. And we've developed very high throughput approaches to measure at high resolution the cellular immune profile of individuals. And we use the platform and the data to address a range of problems in precision medicine. But in parallel, by sort of solving the scalability problem in immune profiling, we are systematically and comprehensively profiling immune health to try and understand how it dictates health outcomes across the spectrum of human disease. And obviously, in terms of the next potential omic, I think it would be a terrific addition to the already very impressive work that's gone on with UK Biobank, and credit to all the hard work from a great number of people here today, and of course the altruism of the participants. But you know, one of the challenges we see in proteomics as it applies to immunology is that you're sort of essentially measuring the exhaust of the immune system. But if you focus instead on the cells, you can build a much richer picture of what's going on. You can sort of look at the engine, if that's not too tired an analogy, and really I think that's where the opportunity lies.
That's fantastic, and I love the analogy about the exhaust of the immune system. Aleksej, Rory alluded to the Elliptica data earlier today, but just very, very briefly. So I'm wondering if you can describe that data to us a little bit more, and also, you know, something that Claudia mentioned, of course, is that there is no gold standard for proteins. We are getting different valuable information for each platform. So I guess tell us a little about Elliptica, and also why you feel this is, again, a good benefit given the proteomics data that we're already measuring.
Thanks so much, Melissa. Just to mention, I'm a co-founder and the chief technology officer of Elliptica, and Elliptica is an early-stage company that uses mass spectrometry to profile UK Biobank. We recently profiled 50,000 samples from UK Biobank using mass spectrometry, and basically, what mass spec is a complementary technology to existing platform in a way that it is untargeted. So basically, what we're doing, we're not just measuring proteins, we're actually measuring everything that basically flies in the mass spec, and then which allows us to get the kind of sequence level information, and that also allows us to resolve the isoforms, different post-translational modification, but also sequence variants, which basically inaccessible by other affinity-based technologies. And when we measure that at the scale of over, in this case, 50,000 samples, now we try to kind of now map all these high-resolution molecular features to phenotype, and also to link to genetics, lifestyles, and diseases, which basically will eventually kind of lead to do the to predict the clinical outcomes. But basically, in summary, the mass spectrometry, it's not just kind of redundant. It's not redundant technology, but what it does is it brings, I believe, that this biological resolution to proteins, and by allowing kind of to assign for UK Biobank to not just kind of like look at the protein levels as they are, but more bring to protein biology in order to understand different molecular pathways and eventually design new or identify new therapeutic targets.
Fantastic. And I think we're all just still so amazed and excited about being able to do mass spec on this scale of samples too. So it's just going to be absolutely great data. I realize we are running quite close to the tea hour, so I guess I want to pause here and see what questions we have from the audience. And obviously, we've got a number of handheld mics. Okay, I think we're going to number 4. Go ahead.
Yeah. My name is Sergey Sokolovsky. I'm from Birmingham. It's Aston University, particularly it's Aston Institute for Photonic Technologies. Don't think I'm a photonologist, I'm a biophysicist mostly. So I do some stuff for my physics colleagues there, specifically for laser physics. But what I'm interested in the talk, specifically your Claudia, and what you said, Aleksej, when you come from, let's say, proteomics through the from metabolomics to the genomics to identify gene, living outside, let's say, the noise. How can you do it? I mean, really, because as you said, Claudia, in the beginning of your talk, everything connected to everything. So it's a question, how do you do this? Thank you.
Well, for me, the answer is very clearly using genomic approaches. So the specificity of the mechanism we see is because we don't start by association, but we start from the genetic signal that connects these diverse layers. So it's extremely specific. And not only that, we can test whether this is specific to a given disease or anything else, any other disease for which we have genetic information. So the absolute crucial part to make sense and move from association to causation is by integrating the genetic information, which is now not just for common variant, but rare variants available in UKB. So that's the answer, and that's the huge delivery that this study enables.
I will just maybe add technically, basically, because with mass spec, we are actually not just measuring a sample, we're measuring millions of spectra per sample. And that also kind of allows basically to train these unsupervised AI-based models to uncover the particles that are clearly kind of repetitive and systematic out there.
Okay, let's go to microphone number 1. Sorry, I thought we did. Sorry, I can't. Do we have a question back there?
Yep.
Okay, good. Thank you.
Hi. Thanks for the speakers. That was really interesting. My name is Yang from University of Oxford. So, Jeff, you mentioned the Alzheimer example quite fast. Can you just elaborate a little bit more?
Yeah. Thanks for the question. So, the specific example there was there's this tau protein, which is a very strong biomarker of Alzheimer's pathology, and I think it's been possible to measure and studied a lot in CSF for a long time. But when you try to ask, can you measure it in blood, it becomes complicated because some of it is, a small amount is from the brain, and then some more of it is basically coming from the peripheral nervous system and is not a relevant biomarker. And so the key thing there is, can you find ways to measure the brain-relevant subset of the protein in blood? And there's lots of work that has been done, none of which I have done. I'm only a student of it. But you can either look at specific phosphorylation forms or at specific protein isoforms. And if you kind of put those things together and you design your proteomic antibodies to really try to measure just the brain-derived subset of tau, and if you have a technology that can go down to like 10 to the minus 16 concentration, because there really isn't very much of it in blood, then suddenly you can get this really, really amazing biomarker of neurodegenerative brain disease in blood. And that, I think, is, presents all kinds of interesting opportunities, both for that specific protein and also for a longer list of proteins from brain or other organs where the same thing is true. And I think technologies like the one that I alluded to, that focus on measuring those very low abundance proteins, I think, are a very interesting complement to some of the stuff that's already being rolled out at scale in the biobank.
Yeah, absolutely. I love that example just because it highlights the ability to translate from a tissue like the brain to blood, as well as why we do find so much value from different technologies of omics. Microphone number 2, and then I think we'll probably have that be the last question since we are running a bit into the tea break.
Yeah, Dave Curtis, UCL. Coming from a clinical background, I think a lot of us would take the term precision medicine to mean having a sort of very precise way of targeting a particular treatment, a particular patient with a disease, another patient with the same disease, we target a different thing. And I'm really not hearing anything that makes us think we're approaching that or getting towards that. And am I being unfair, is my question.
Who wants to take that one? I think, Dave, it's a fair enough point. And some of the stuff I showed, for example, in the clinical translation is mostly about finding people for whom we probably already have a recommendation and just trying to make sure they do it. Change your diet, stop smoking, maybe take a statin. I think that's still worth doing, but I sort of take it, I think the fair point is that precision medicine, maybe not. Anyone else want to?
I'd be very interested to hear your perspective, Slave. I think I was fascinated by the genetic resilience component. And I guess my question is, can you envisage a future where each of us is taking our own cocktail of prophylactic agents to paper over our own genomic cracks, if you will?
Well, I mean, I'll take a different approach to the answer. I think it is a fair point, and most of the presentations here were more around precision diagnostics than precision medicine per se. But I think also we shouldn't think about precision medicine as a tool for every single indication. There are clearly indications where it is very important, very relevant, especially in the context of monogenic diseases, but even some of the common chronic diseases. But for many diseases, actually, you're probably better off developing therapeutics that can benefit the broader patient population by targeting an underlying common mechanism rather than a precise gene perturbation in that patient that's driving that patient's disease. And we're recognizing this increasingly, that if we look at across these common complex diseases, they're rarely caused by one particular perturbation, one particular pathway that's perturbed, but they have similar clinical manifestations. And then it's about how do we identify the patients that will benefit from a particular intervention and then target the therapeutic options to those right patients. I think the extreme that many people think of around precision medicine is more around therapeutic gene editing context, and you're precisely right. We didn't touch on those points today.
Yeah, that's absolutely a fantastic answer from both speakers. I think we are well into the tea break, so I'm going to call the session to a close, and obviously feel free to come find any of us during the break, and thank you so much.